Today we are going to focus on scientific communication, which is our today's theme of the 3-day international colloquium. We will have two talks for today's session. The speakers for two talks are Professor S. Ganesh, Deputy Director IIT Kanpur, and Dr. Ipsa Jain. Thank you so much, sir. And I think ma'am also have joined for your valuable presence, and we welcome you. Before we start the session, it's humble request to audience. Please keep your mic off at all times for better bandwidth. Type your questions in the chat box. They will be picked up end of the talk. So now I would like to invite Dr. Shashi Chawla, teacher in charge, Microbiology Department, to introduce our guest speaker to the audience. Over to you, ma'am.
Thank you, Dr. Indamani, and a very warm welcome to all the participants and speakers. So we all know that communicating science clearly and concisely is important so that it is understood right from peer-reviewed articles to blogs to tweets. Effective communication are becoming part of the core professional skills which every science student and professional should have. And this helps not only in getting research ideas funded, but it also in becoming a better teacher or mentor. It is equally important that science is communicated effectively beyond peers to broader non-scientist audiences as it builds support for science, promotes understanding of its wider relevance to society, and encourages more informed decision making at all levels, from government to communities to individuals. So, in this light, we are fortunate to have Ganesh with us, and it's my honor to introduce him as our keynote speaker, who would be enlightening us on the scientific communication. So, Professor S. Ganesh is currently the Deputy Director and P.K. Khelkar Endowed Chair Professor at IIT Kanpur. He has also been the Dean of Research and Development from 2017 to 20 at the Institute. Professor Ganesh received his PhD from Banaras Hindu University and his postdoctoral training from IISc Bangalore. He worked as staff scientist at Riken Brain Science Institute, Japan, before joining the Department of Bioscience and Bioengineering at IIT Kanpur. He is an accomplished researcher in the area of molecular genetics and neuroscience. He works on genetic forms of neurodegenerative disorders in humans to understand their genetics and disease mechanisms and to develop therapeutics. He's been serving on the editorial boards of journals and has several quality publications. He has many accolades on his name, including prestigious Professor J. Das Memorial Award Lecture bestowed by the Indian Society of Cell Biology in 2019. He is highly recognized and appreciated for the MOOC, uptaken by him under the National Program on Technology Enhanced Learning, an initiative supported by MHRD, Government of India. He's been taking courses on functional genomics, human molecular genetics, different aspects of professional scientific communication, including written communication, oral communication, reading, and ethics in communication. So with great privilege and immense pleasure, I invite Professor Ganesh to address the audience on scientific communication. Over to you, sir.
Yeah, thank you, Dr. Chawla and Dr. Tyagi for inviting me to give a short talk in this colloquium series. This topic, really requires a lot of time. This scientific communication is something that if you have to impart, because it's like training that I'll be talking about. So it can't be covered in 50 minutes or whatever. But what I'll be doing is a kind of a highlight as to how one should plan to be able to communicate better. I mean, this is what I'll be touching upon, and I'll give some references and other links where those who are willing to learn more and practice and get better can really access to those additional resources, and you can further your skill. So it's a great opportunity. I always I love talking to the students. I have been teaching in this IIT Kanpur, even the core courses, that is, the first year undergraduate students. So just after their class 12, it's a challenge to talk to them, especially when they join IIT and when they don't like biology. Majority of them hate biology. And still, our job is to make them like biology, and we found many of them switching their field. And some of my own colleagues now, they're former students, and after that BTech, went on to do biology, either in computational biology or switched completely into hardcore biology subjects, is because we are able to communicate how interesting biology is a subject. So communication is something that is very, very essential for day to day, your interaction with anyone. So that's the one. So let me share my screen. Able to see my screen?
Yes, sir.
Okay, good. So that's the topic, scientific communication. Go on further, I would like to tell you that I have a series of lectures on the same platform, the NPTEL platform. There are about 20 lectures that I have just listed here. You can just do a Google and YouTube. You will have this free access to all these lectures. So what I'm going to discuss today is something, a bit of summary of this 10-hour lecture series. Because not everything can be covered in about 40, 50 minutes. But this gives an idea as to how important and how you can further your knowledge through communication. Um, just to give you an idea, like each one of them that you see on the screen are artists, right? Whether they are, um, vocalists or musicians or artists or composers or dancers. Each one of them have become masters, a legend in their field because of one, the interest they had in this field, and number two, they have really trained very hard for years and years to perfect the arts and to become masters. So what is very clear is that one has to spend a lot of time to really upskill your talents. Even if you have that initial talent, then you have to really make it better. Therefore, you become one of the legends in whichever field that you choose to become. No exception. Here also, it's not that day one you will be able to communicate better. Even if you have limitations, with training, with putting in a lot of effort and understanding how you can make your communication better, you'll be able to become a very good orator, speaker, or writer. That's possible. It's not that I was fantastic in my teaching abilities when I joined here as a teacher. But with time, I learned exactly what the students need and how it should be conveyed to them. So you become an effective communicator. So it's a lifelong process. If you ask me whether I have perfected, the answer is no. It takes time, and as you can see, that there are a large number of research articles written as to how you can enhance your skill set. So that is a long process, but you have to put in a lot of efforts for this. Really common because if you look into the various kinds of media that you have now, whether it is the print media, whether it is social media, any of these media, then you certainly have different ways of communication. For example, Twitter, if you talk about, it comes with a word limit, and you should be able to express within that word limit, right? So all these are, ways of communication. It gives you a tool for you to express your thought process, not necessarily to forward a message that you receive from somebody. So if you use it effectively, you can, even in day-to-day life, that you come across this kind of platforms will help you to become better, right? So if at times we think that writing a story is not that big deal, but if you attempt writing, you will understand that that it becomes very difficult. You don't know where to start, right? That's that the very first line you will not be able to finalize it because it's a challenge that you thought that is very simple, but it does take time and effort, and it's not easy to begin with. But with practice, you'll be able to do. And scientific communication also is not something very different from storytelling. If you are very good with storytelling, the same kind of a philosophy can apply to scientific communication as well, except that it is not imagination, but based on facts, right? That's the difference. But the way you are able to put across your point, the basics, the philosophy behind the communication, even in science, is very similar that we will touch upon in a few slides from now. So, but if you look at what is communication, right? It is basically, as an individual, I look at something and then I codify it and then, convey it to somebody. The other person is able to decode, right? What do you mean by codifying and decode? Suppose I touch something which is very hot surface. I'm going to tell my friend that don't touch that place because it's very hot. Now, hot is something, a word in the language English, but that's, only if you know the language, then it gives some meaning. If I do not know the English word or English as a language, if someone says it's very hot, I'm not going to get anything. It's just noise for me, meaning some sound. In fact, all the languages that speak have evolved from, our ability to make different kinds of noises and attaching some meaning to that. So that becomes a word, and then you have the grammar to construct sentence, and then it is being told to you. You learn as you grow up, and you're able to decode it. That hot means something, something that's going to hurt you if you touch. That's how you learn. So if you are unable to code it, or if the other person is unable to decode it properly, then you are unable to convey what you wish to convey. That would be a limitation, especially when you interact with the society. The society could be, your peers, your evaluators in exam, or you go to an interview, or you make a presentation of your work. Wherever the time and effort that you put into doing a such topic, it becomes less meaningful if you are unable to convey what contributions you have made in the research project that you took. Okay? So that's possibly the reason why we have to learn how we can, equip ourselves to communicate better, right? So it could be the way you speak, or it could be written form, or it could be visual communication. But that's how you do. And when you do communicate, then there are going to be questions. With the questions, you would understand whether you are able to put across the point properly. So that's where you call it feedback. And they don't get, when the audience do not get the meaning of your presentation, then you would understand that you haven't made a good presentation, right? So that's how you learn with time how you can make yourself better. So there are three major forms of communications: verbal, non-verbal, and visual communication. This is common for all communication, but in scientific communication, the first and the last are more important than the middle one, the non-verbal. We'll see what it is. In the verbal, of course, it is oral, the way I'm doing. I'm making a presentation and I'm speaking on the topic that is given to me. That is oral communication. Written communication is something that you write a paper or write a newspaper article or write a thesis. That is written communication. And the non-verbal communications are expressions, basically the way you express, you greet or you show your anger, or it could be the theater performance like Kathakali or any other, which doesn't necessarily need to go with certain words. It's a spatial expression, but that's the non-verbal. But the visual communications are very common. You see every day as form of the signages in the street, or even if you go to any mall, that you find that there is restroom, or if there is anything that one should not touch, there's a danger mark. Or in the COVID times, you find that wearing a mask becomes mandatory, and there are signages for that. So this is independent of the language because now it becomes kind of a universal symbol wherever you go in India, regardless of which state you are. The roads will have the same kind of signage. You don't need to know the language, local language, still you can understand. There's going to be a bridge, there's going to be a school, a hospital, and so on, right? So that's where it becomes very, very important communicating because you don't know who will use the road. So whether he or she may know a given language or not, you don't know, yet you want to communicate, right? So what is scientific communication? So far we have seen the general communication. So as I told, oral communication is giving a talk or writing a paper that comes in such article comes in a journal or writing a book or thesis and so on. And the visual communication is, giving a complex pathway like what you have seen, citric cycle or anything. Or, all the illustration that you see in a book, again, these are all visual communication, and you have, a celebrated speaker next session where she'll be able to, Dr. Jain will be able to do more on that, so I'll not touch upon this topic. But that's equally important. And how about non-verbal communication in scientific communication? It's not something very, very essential, but certainly would like to smile when you speak. You don't want to keep an angry face. Again, that makes you a friendlier speaker, friendlier, teacher that where people can approach and ask you questions. So certainly that helps. Welcoming the audience to ask questions is one of the good qualities of a good speaker, right? That's again is part of it. These are the topics that normally is covered from developing an hypothesis, I'll touch upon, to presenting your work to a common man. That pretty much comes within the scope of scientific communication. And of course, this is aided by your illustrations, your data, and depending on to which audience you are speaking to, right? So all these, whether it's illustrations, schematic, figures, bar diagrams, line diagrams, this is to convey your findings in a simpler way than the raw data and we give an analyzed version of it. Therefore, the other person can appreciate what you have done and maybe take it forward or even question what you've done. That is what the research is, right? I'll touch upon the hypothesis. It's very, very important. I'll tell you why a little later. But when you look at any research topic, the first question that people would ask you is that, you take up a MSc project or a PhD work, you have to find a problem, right? So you have to find a research topic to work on. And often this research topic is something that is a question, but nobody has found an answer to it. That's what you take up as a research project, right? The question is like, you have to ask questions. And you need to know what is known so far. Then you have to ask questions. And the research is normally divided into two sections. One is replicating or extending the work. Like, for example, if you join a lab which has been doing research on a given topic, the senior student would have done some work, and that led to very good understanding of that subject, but then maybe a few new questions that have come. So that you need to work on. For that, you have to replicate some experiment that senior student has already done. So that's where you begin, and then go on to do more experiments to answer unanswered questions. And the other one is, therefore, sometimes you have to redo what others have done to check whether indeed that's the case. I'll come to that, why it is important. In research, replicating others' work is as important as doing something new, because replication, if whatever you have done, if others are able to do and show very similar results, that means you have done a fantastic work. Replicating what you have done is as important as doing something original. And original is something, of course, you start on a new topic and then go on to do. So it all starts with the kind of questions. And you need to ask questions. How does this work? Who says so? Things like that. Is that true what you're saying? So let me test it. Why are you doing this? why are they both right? If there are two different possibilities, can we learn from abnormal? This is what very common in biology, right? Especially if you look at mutant that we talk about in any organism, or creating a mutant. That's how you understand what is a normal process. That is by creating abnormality. And of course, rare condition, that uses more opportunity. Even in disease aspect, for example, people talk about old age disease like Alzheimer's. Again, there are rare forms, these are genetic forms, but there are common forms that are because of something else, environmental influence and so on. But the rare forms really gives an insight. So these are the ways by which people take up research questions and do more studies to understand what goes wrong, right? So that's how you go about approaching the questions. Like these are the questions, right? What, where, when, how, why, who, these kind of questions. Who meaning which player? What is the factor? Why it happens so? When you remove this gene, why the cells don't divide? Things like that. Now, how does loss of that gene affect cell division? When does it affect? Where does it affect? So if you use these questions, you'll be able to build a research project. That's very important. And that's what is called as inquiry wheel. So you have certain observations. Based on the observations, sorry, you define a problem, and then you ask questions. To address the questions, you come up with some methods. That's what called as investigation. And then you say, if I do this, I expect this or that. And then you do the study, you get results, and then you reflect on the findings, you give conclusion, and then you communicate to the scientific community or the society. When you do so, it's not that you will answer everything. There will be many questions that are answered. There will be new questions that will become the basis for another round of projects or theses or whatever you call it. That's why the inquiry will continues to go. That's what research is. Lead such, keep doing again and again. Just to give some examples. For example, if you want to really look at ideas where I can get to do my research topic, the newspaper are good enough. During last 1 year, during the COVID pandemic, you have lots of newspaper articles that talks that this, for example, the virus that that causes the COVID has leaked from the Wuhan lab. Some even said that this man-made virus is not that something that is native, as was told. Some other newspaper articles said this has come from what you call as some animals, called as zootonic or whatever, and then things like that. There are multiple theories, and also say that everything is possible. Everything is possible because every option is not yet proven otherwise. So until you prove that is correct or not correct, the possibility exists. I'm sure those students from this department can understand and appreciate this better. This is how you come up with hypothesis. The hypothesis could be it's a man-made virus, or it's something that was being studied in the lab to understand how the virus functions, but somehow escaped the lab conditions that were studied in the population. Or these are some of the viruses that to begin with, the animals were the host, but because of the humans consumed some part of the meat from these animals, it jumped into humans, or humans went very close to these animals. These are the possibilities, but you have to go with some observation, some basis, and then propose that as an hypothesis, and then prove. That's where the project begins. So that's where the hypothesis is extremely important. So how do you build hypothesis? There are two ways. One is called as inductive reasoning. You have some observations, and that observation gives you a limited set of patterns, and based on the patterns, you come up with a hypothesis, and hypothesis becomes very solid, and then it becomes theory. Okay, it remains as a theory because some of the hypotheses cannot be tested. For example, evolution. When you talk about evolution, is well accepted that we all evolved from primates, monkeys. It's accepted concept, but it's not. It's proven. It's not. You can prove because it's a more than a lifetime observation that is not doable. Therefore, it becomes an exceedingly attractive theory. It remains a theory, but that would be hypothesis that can be tested, and that's what goes to what you call as you do experiments and then you have observations with that you confirm. So these are the two different patterns: inductive and deductive reasoning. And in inductive reasoning, you look at the pattern, for example, what is the last square? It will be the same kind of hourglass. You can say that is based on certain pattern. That is a process where a small observation is used to infer a larger theory without necessarily proving it. It is based on theoretical assumptions. And that's what you do, in inductive reasoning. But once you have a tentative hypothesis or theory, you can do, in some cases, you can set up experiments to confirm, test the hypothesis and confirm, then it becomes the knowledge, right? That becomes the fact. And that's how the textbooks are there. When we talk about mitochondria, cell wall, whatever we have studied, the biochemical pathways, these are all based on this kind of observations that you come up with, the facts, and then give a meaning to that, right? So that's what you do. Induction is a possibilities. Test the possibilities by setting up experiments and prove that, and that becomes the truth, right? So when you talk about meiosis or mitosis, we know that DNA replicate and then cell divide. Again, you have the same number of chromosomes. That is now a fact. If it becomes reductional, it becomes half, that is meiosis. And we know that indeed it happens because that is again proven to be correct. So that's become the truth. That's what you have in the textbook, right? This is how the whole cycle goes on. Now, why are we talking about communication? Because you learn from the experiments that people have done, 100 years back. When you talk about, when you look into any of the textbook, the very, very word cell has come from some observations. I don't know whether you have read in textbooks, somebody took the cork from the wine bottle, made thin slice of it, and then looked at under the microscope, and they found beautiful, kind of cells. Cell meaning prison-like, different rooms, and that's how it was called a cell. And later we know that in the wood, nothing but the cell wall remains that word. It looks like that. Or the X chromosome is something that is named after, when they stained the nuclei of cells derived from a cat, female cat, they found something which is very, stained very differently, only the female. And the cell derived from the cat, female cat, they call it an X. Later we know that is nothing but a condensed X chromosome. Therefore, that chromosome was given the name as X. So this goes on like that. To make observations, that's there in the literature. You go and do more studies on it, and then you'll be able to find what exactly it is. Therefore, recording is very, very important. That's what it talks about. You have an hypothesis based on certain observations, and the observations are such that it is good enough to form an hypothesis. The hypothesis is very attractive, a possible kind of a answer to a question that is not proven yet. That's what you call a hypothesis. And then you go on, testing the hypothesis using certain methodology. It could be experimental biology. It could be even a kind of population studies, depending on what questions you're asking. And then you have your observations, and then you interpret, and then you make that as a finding. That is what you call a discussion. So this is the flow of any research project. You have a hypothesis, and then you have well-defined questions that you would like to test using certain experimental methodology. And when you do that, you have data that is called as observations or results. And then you interpret the result. What do they mean? You give meaning for these observations, and then you extrapolate, with other knowledge that you have from the literature otherwise. You extrapolate, that is what called as discussion. But this is what in the form of writing. And as you can see here, I introduced already that you are artist. each one of them doing a different performance. Some are singers, some are they play music instrument, but you must have seen someone like this. This person does everything. He has to beat the drum, he has to play whatever it is, and then sometimes they sing as well, right? They have to do multiple jobs. And unfortunately, in research, it is something like that. You have to read, come up with hypothesis, and then define the problem, and then come up with the methodology, conduct the experiment, make all the observations, infer, write, and communicate to others. So just being a very good reader, knowing everything doesn't help. You have to comprehend and ask questions, critical questions. When you say ask critical questions, not always you have to ask someone else. You ask yourself, then to answer it, you come up with methodologies, test it using experimental design. So you have to perform the experiments, and then you analyze the results. And the analysis includes multiple ways of doing it. It could be statistics, it could be image processing, it could be anything. And then you infer, what do they mean? And then you discuss again, you go back to the literature and write it, and then convey this to others, both in the form of writing, or you make lectures, or defend your thesis and so on. So you have to be good at everything. Just being good with one is not going to help you. You know everything, but you're unable to convey it. You may not pass the exam. So you have to have all the abilities like the person that is shown on the screen. So that's where it becomes challenging. And that's where you have to learn something that you are not very good at it. Okay? If you're unable to read, you will develop the habit of reading. If you're unable to communicate, then you have to develop that ability as well, whether it is oral form or writing or whatever. It's very important. Right? But again, hypothesis is important because unless you introduce to the audience what is the importance of your study, why you have gone about doing this particular project work, unless you drive home that point, they are not going to sort of appreciate what you have done. So that's where the hypothesis is extremely important. That leads to a very good project. Okay. Let's go back and ask question, what is research? When you ask this question, you ask to your parents or someone who is not directly in research field, and then or you close your eyes and ask this question, what is research? Often this is a kind of image that comes to your mind. Some lab, something like a Hollywood movie or sci-fi movie kind, where people are there in the ground, like more often in COVID times, you must have seen everyone going with this kind of a PPE kit. That's become a common thing when you talk about research, right? And you can see that in many of these commercials that you see in the TV or in the print media, you find that someone is in the white color coat, right? It could be a drink, energy drink, or it could be a toothpaste, or it could be as simple as a bleach. It comes with different trade names, right? Just to clean the surface, right? All these, will have somebody who's having a white coat. Could be a doctor, it could be a scientist, but that's what it is. Why? Then many of these commercials have this kind of background. Why do they wear white color coat, and why do they show a lab like images at the background? Is because an important thing is that this is something that is a truth. That's what I told, right? The research structure, the way you do research project, allows anyone to look at what you have done and then validate it. Right? Indeed, that's the case. You show everything what you have done. Therefore, it is quantifiable. Meaning, you're not saying that this detergent will kill 100% of the microbe, just like that. You have done that analysis, right? And then you have quantified it. So there is a data to tell that the detergent really works well, or bleach really works well, or talk about detergent that it removes the stain. It can quantify. Verifiable, meaning you can give it to anybody, the data, and they can go through and say, yes, what you're claiming is correct based on this data. Or they use that, your methodology, and then they can do this experiment again. Therefore, it's called replicable, meaning they can reproduce. defensible, meaning your conclusions can be defended based on the data. That's where the reliability of the data goes up. Many more researchers, if they do the same kind of work that you've done and they're getting the same result, that gives a lot of kind of credit to your work, because they're able to others are able to reproduce what you have done because you have given all the conditions and it works fantastic. That's why any scientist saying, people take it with confidence because they don't rush with the conclusion. They do all the controls, they do the quantification, they are certain about it, only then you convey it. That's why there is a credibility for any scientist, and that's why all these commercials that you see, people represent with the white tips saying that he or she is a scientist, and that is because of scientific observation that we are claiming. Whether it is indeed the case or not, is a different discussion point. But therefore, documenting and telling what you have done is extremely important, and that's what communication is when it goes to written communication, and that is what is called as hourglass notion in research. That is, begin with broad questions, right? You may ask, for example, for organisms to survive, the COVID-like situation, then they have to have certain pathways to fight against the microbe. Let's say, I'm just giving you some hypothesis. When you say organisms, right, like in an infection, then eventually you will not be able to using an organism. You may use a cell line, but that's the within broad questions and narrow down and focus. What you do is you look at a specific event and then use a model system. It could be an animal model or microbial model, that E. coli as a model, or it could be cell lines. So you narrow down. And then you execute operationalize, meaning you do the experiment and then you observe. So it goes like a hourglass. The observations are very limited. And then you analyze the data. And then you reach conclusions and then again generalize back to the question. Again, you apply your findings and conclusion again back to a larger context. That's where it's called as hourglass, because you can't do experiments with a large number of organisms, only limited species, what you call as a model system you can do. Or there are various other limitations. Therefore, always that is very, very well-defined, question that you ask, but the outcome could be of, larger consequence. That's how you do. Like if you look at the cell biology book, most of, what you talk about transcription, translation, classic textbook, how come from yeast as a model system, but it doesn't really say so. We believe that's what's happening in most of the eukaryotic system or advanced organisms that you see. So that is a generalization that always there in the science, right? So the first one, the first two, begin with a broad question, narrow down. That's basically based on the hypothesis. You have to have a very good, solid hypothesis. Then the rest of your experiments are pretty good, and that's how you do, right? Now that's for doing the experiments. Now come to how you do writing. Right? This is one paper, research paper. After 4 years, 5 years of your research work, and let's now look at how normally this is being written, right? So again, this is what I said. This is a hourglass model. So you have an abstract of that. There's a summary that you have. You can see in the screen there is called something called abstract. That is a summary of your research finding on the paper. You read that, it will tell exactly what the paper is about. Then you have an introduction section that gives you the reason as to why this study was done and what is that is lacking in the literature and why it is important. That's what introduction is. Then you go on to do methods, tell exactly what you have done, how you have done. Results are observations, and then again you extrapolate your results to generalize what you see, larger context. That's what called a discussion, right? So how do you really do? So this, the structure of introduction, methods, results, and discussion is called the INRAD, sequence, right? That is the traditional method of writing any of your findings, even in your thesis or the papers, research papers. That's what people follow: introduction, method, results, and discussion. At times it could be introductions, results, and discussion. Methods may come a little later. There's a possibility. But traditional, journals will have this sequence, and thesis invariably have this sequence as well. So that's a standard thing. So let's quickly, touch upon about the topic. Like, for example, introduction of the topic. That's what introduction. So you basically give a hypothesis, define the question, then you tell how did you test that, or answer your question, that is study design, and your observations, and then what do they mean? That is the interpretation, and then finally discussion, right? So, this is what you're going to write. How do you write? How do you start? And what you say is something very, very important. Otherwise, the reader will not read. For example, you read this, two sentences that is given. I looked at everywhere, from this corner of the room to the other, even below my desk and car, but I could not find the pen that you used this morning in the very same room. So until I come to the second half of the sentence, I really do not know why this person who is writing is looking everywhere. It's not very clear, right? So, that's something that one has to, in research, you have to say why you are, whatever you have done, you say that first why you should do that, right? Therefore, you say that, for example, I lost my pen, and then you say you search everywhere to find. So that's the concept. If you go to my YouTube lectures, that I have given link to many of the websites where you can practice this. So there are three important elements in writing, or even when you speak. First is given and new. So always when you start anything, you start from somewhere where the audience already know the topic. That's called as given. And then you go to something new that probably they will not know, right? So you start that. That progression is extremely important. If you start with something that they don't know, then they're going to give up. They lose their interest, right? So that's not going to help you, because your main job is to convey what you have done. So it's important that you keep the audience with you, you speak. Therefore, you always want to build from something that you expect all the audience to know. And then call as creating the focus. You from the larger context, you take them, you narrow the question that you're asking. You help them to reach there. And then that is called as a topical progression. In this process, you also have to keep in mind that you always tell things that are light, meaning it's not that complex, and then build your complexity, right? It's very important, because then only you can keep the audience with you. It's just like, that's why I told you storytelling, right? If you listen to your grandma, the storytelling, they always start with long, long ago there lived a king, Anybody would be able to relate this. And then they go on narrating, with, the other complexities of the stories, but it's easy to follow. Exactly, very similar concept. You will use a good paper that you feel good to read. That's something that you need to practice. I've given several examples in the YouTube lectures. You would like to go through them. And that is what reflected even in the abstract of the paper, right? The abstract of paper is nothing but, a summary of the entire work that talks about the background. It gives you the hypothesis. It talks about the method. It talks about the results, and what do they mean in a larger context. So that's what it is, the abstract is. So, but you should know that abstract is written at the end of writing the rest of the paper, not before. It's the last one to be written, right? The question is, who reads the paper? Because you write the paper not because you wanted to write. Like, for example, A.R. Rahman, or anybody who sings, for example, Arman Sami, anybody, because they sang so many songs for others, not for themselves, because they are professionals. Very similar way, when you are a scientist, you do research and then you communicate for other people in your field to know what you have done. Therefore, you have to make sure that your papers are read. So how do they read? How would they come to know what you have done? That's where, if you look at the journals, they have what is called a table of content, right? The table of content gives you the title of all these, articles, and if you look at the um, the readership, you can find that the title has got maximum followed by abstract and then only the rest of the papers. But if you go to the rest of the papers, because they may not be interested in the details, they may not know what you have done. Only those who are interested in the topic will go down, right? That's why the title is extremely important, because they should be catchy. It should attract the audience to read the abstract, right? And it's very commonly used in commercials also. They're very, very catchy, right? It sort of conveys what, really. For example, Domino's, 100K. So that's something that, entices you to go for it, right? And a good title brings visibility to the paper, because that's how they read your abstract. That's how then they read your paper. It's extremely important. And, there are papers on that, how to write a good title for your research articles. Again, you can understand that, why it is so important to learn to write even a good title, because that brings in more visibility to your work. And this is what it is. Articles with results describing titles are cited more often, because when people want to say that very similar observations were made by others, they're not going to read the entire paper. They'll look for titles that have very similar findings. By giving some such, we'll come to that. And then you read and then cite. So obviously, if your title is not something that conveys what your research is not going to be read by many in the field. So it's very, very important. And that's why the title, and most of the journals give you the guidelines how to write a good title, right? That's one way of getting to the article, but there are other ways by which people look for similar kind of work. That is by doing search. You must have seen PubMed is an example. In biology, you give some keyword like epilepsy and such. It lists you large number of papers, right? But that's many of the papers will not have epilepsy as a topic in the title, but that's something it deals with that subject. So that's why, for example, for this particular article that's put in here, there are keywords that they call. As an author, you make these keywords, and these keywords are something that anybody is interested in knowing any work in, say, for example, leptin. If they give such this word, then they'll be able to get to this article. But what is keywords are something that you should give something that is not there in the title, because title also can be picked up by these, for example, leptin, which is anyway there in the title. So if you want your paper to be read by many, you come up with some words that are not there in the title, but again related to your work. So that is how you give the keywords, right? The title is important, keyword is important, because you have a lot of data in the public domain, and this is how they are able to reach with that. Maybe in another 5 minutes I'll conclude. I'll give you an idea what it is. The title, abstract we have done. So how do you do the rest of the section of your your project or report or your paper? This part, introduction, method, results, and discussion. So the way the order is done is you write methods first, then you go for results, then you write introduction, then you do discussion, then abstract, then finally title. This is the normal way of writing research paper. Generally, people don't start with title and end with discussion, because it's difficult to write. So you can see that there are papers on that. If you ask, for example, a doctor who is in mode, how do you really fix the body, right? Embalm the body, they'll say it's very easy for me to explain than do it than writing. It is a challenge, right? And same way, if I ask you what is the color, how does black color, can you explain? It's very difficult, right? So therefore, it is harder to kind of express what you see or what you observe, but you have to learn how to do it. It's very, very important. So the way it is done is that create regular time blocks. You want to submit your thesis, say June is when the convocation is that you want to submit in May. So you put your deadline there, that May whatever 30th I have to submit my thesis. You work reverse. That is what calendar is. If you're submitting in May, it should be corrected, before that. Then you should be giving to your supervisor. Then if that is the case, then I should be finished. Then what are the experiments I have to finish? You put everything there in the calendar, create detailed outline and discuss it with your mentor. It is important that this is how I plan to, organize my work. Discuss before you write, or discuss with your peers, right? And be meticulous and accurate in describing methods and material, because that's extremely important. Then only others can replicate your finding and they'll have trust on what you have done. Therefore, you write exactly what you have done. Be clear, concise, objective in describing your results. Exactly say what it is. Don't interpret. It is observation. That's what called as results. Interest your reader in the introduction section. So when somebody reads the introduction, then you should generate interest to read further. Otherwise, they'll stop there, right? So you have to somehow generate interest as to why your work is important, right? So by signaling all its elements and stating the novelty of the work, what is that something new that you have done? Present the principles, relationship, and generalization. So you say that it all applies to everything. This may be unique. You make it very, very clear and convincing to saying that this is what is kind of you can apply the results to understand. It's a larger context. That is, and revise your paper once you are done, or you have to go through again and again and make it better. That's what it is. And that's the hourglass. And if you see that, you'll find that in this 7 rules, you start with methods, go to results, and go to introduction, and finally come for discussion. This is the common sequence of writing any research paper or thesis or whatever. This works the best, although that's not the order in the research article, right? It's very important, you create outline and discuss, right? So start with an outline. So how do you really make an outline, right? So take a blank paper and write down in any order all the important ideas or questions that you have asked in your work, right? Once you are done, like, what is the topic of my paper? Why this topic is important? How could I formulate my hypothesis? Then you rearrange them in the logical way. That's what I told you. Less complex to more complex. Topical progression, light and heavy, and then things like that. You make that, and then that's how you write, right? And these are the points that you put together. Like, why your research important? What is your hypothesis? What are your objectives? What materials you have used? What is the study design? What are your most significant results? What are your supporting results? And what are the major findings? And what is the significance or implications of your finding? And if you can put them all in a piece of paper like bullets and arrange them in order, that basically forms your research article, right? You can say this is introduction, this is methods, this is results, and this is discussion and conclusion. And once you have this skeleton, now you can add flesh to this and make organism. As simple as that. You have these bullets. In each bullet you start expanding. That becomes your starting point. That's how you write. And that gives you an idea, like, I'm going to cover this part here, I'm not going to discuss that, and so on. So when you know exactly how you are writing. So this is very, very important when you write any research paper or thesis and so on. So once you're done, it's getting feedback during early stages of your draft is very important, saves a lot of time. Talking through ideas allows you to discuss with people, outline the most effective way of communicating. You discuss with your friends and seniors who have done that. And then having a list of journals where you want to publish, that's also very, very important. That's where this is, it becomes challenging. How do you start doing it? But if you make all these points, you'll be able to do it better, right? So one simpler example is you have one paper that is already published closest to your work, very similar concept. You have that as a template and see how they are written. You follow that pattern in terms of how you write. That if you're a first-time writer, that really, really helps you. And that's where all this important. revise your paper as many times. And if you look at this is a stone, but the stones are the one that is used for making a diamond. So you have to polish them as good as you can. Therefore, your papers become better. That is called as revising and editing your manuscript. You go on, look at the macro level, whether this point should come first or later, or this micro level is that changing the words and making the sentence more readable, right? Or appropriate words. These are very, very important. Macro structure and microstructure, deal with them separately. And how long you will revise? Many times you have to revise, but when would you stop? That is where it's important. You have to go on, doing all this fine tuning. You stop because you had a calendar that you have to submit. So one day before that you stop, and then that's the best that you can make within the deadline to submit. That's to your supervisor. He or she would work further on it. That is how it is, right? That's how it's done every time that it's done. So you would feel good, right? So this is the process. If you submit an article, what happens to that research paper? It goes to an editor who reads, and he's not going to read, or she's not going to read and pay a paper. The title is read, abstract is read, and if they are convincing, they are sent it for reviewers, and reviewers go through everything. They give a feedback that the work is good, can be accepted, or these are the kind of a shortcomings. You address them and submit again. That is called a revision, or is rejected, right? So it is not that it's rejected because you have not done a good job of doing experiments. It could be that you haven't really written it well. So that's where the writing becomes important, right? Then it goes this kind of cycles to finally get published, and that's all about the kind of research articles. I'll take one more minute to tell you the difference between a research article and a proposal. With that, I'll end my talk. Research paper is something that explains what you have done. The research proposal is something that tells you what you wish to do, right? Which is called as, in some university system, you have synopsis, PhD synopsis, something that you give a proposal, or if you want to go for a funding from some funding body, then you have to do a proposal. So in research paper, you write everything from introduction to discussion, but in proposal, you have not done any result, right? You have not done any experiment. You give only what is the background, what is the question that is there, why it is important to address, and then you say that why it is important if I do this work, what impact it would have. So that's the difference between a proposal and a research paper. A proposal, you say the rational hypothesis, everything. So then you sort of end by saying that if I do this is what I'm going to offer back as wisdom, new knowledge. That's the impact, right? But it goes on. This is a cycle that you have. It goes on. It's very similar, concept that you have to convince the sponsor why they should fund. Therefore, it becomes extremely important that you put a very, very good hypothesis. But all these topics are covered in my lectures. So I would recommend those who are interested can go through these lectures, because it's not that I can discuss them everything. Each one requires a lot of time to emphasize as to how important they are, because they are all equally important. But you can go through these lectures, and I would end here, and I'll be happy to take any questions that you have.
Hello, am I audible?
Yes,
Yes.
Uh, Thank you so much, sir, for such a wonderful talk. It's really enlightened us. So now I would like to invite Dr. Kriti Tyagi to moderate question and answer session. Over to you, Dr. Kriti Tyagi.
Thank you, Dr. Indramani. So first of all, I have to admit I'm one of those who start with the title. I have to start doing it other way. So let's start with question and answers. Uh, so the first question is from our second year student. She's asking how many attempts does it take for one paper to be approved?
I guess if the question is correct, then how many attempts it takes for a paper to get accepted in a journal, or I believe, or um,
I
you know,
guess that's the acceptance.
right. So it all depends. Like you need to understand one thing. Like a paper, even a good research paper, a good paper that talks about a good research work may not get accepted in a journal if that journal doesn't have that kind of work. So when you do an experiment, you write a research paper, you should know to which journal you should submit. So there are focus areas, aims, and objectives of the journals as well. So you have to first choose that correctly. Because if you do not choose that correctly, then it will be rejected. Second, how rigorous your analyses are, how rigorous your methodologies are, and what is the rank of the journal? Because in every field there are top ranked journals, middle level journals, and lower middle, and there are journals that wouldn't accept any work that is done reasonably well but may not have much significance. So there are different categories. So you have to aim for appropriate. Like for example, it's not that all my publications are in good journals. There are some of my publications are not so in great journals. Um, I know that it will not go in higher journals, so I should be able to pitch, right? So if you do it correctly, if your hypothesis is correct and your methodology are appropriate, and you have written it well, and you have chosen an appropriate journal, you should go through, right? But if you don't do any of this correctly, until you find an equilibrium where they will accept yours, as because every time when you submit, the referee would give you some inputs. You may improve it accordingly. So with every attempt, your journal paper also gets better, and you may find it. But you should, if you know where it will go on, if you submit, uh, there's a 50% chance it will go through. In the sense, if your Plan B journal would go, and certainly it will go through. If not Plan B journal, that's why I'm saying 50% chance.
Thank you, sir. I have personally one question, and a lot of time we get confused with that term science communication and scientific communication. Are they same? Are they replaceable, or there's a difference between them?
It's a difficult question for me also. I don't know. I think science communication is something that communicate about science to the general public, if my understanding is correct. The scientific communication is that you are communicating to your peers, like for example, journal articles, proposals to the experts. The science communication is possibly, I'm not too sure, but I'm just making it one thing. Okay, yeah.
Thank you, sir. Our question from Gargi Samasta. She's asking, every scientific experiment has some amount of uncertainty. So how a person can reach to 100% truth about the topic? I guess she's talking about, the assumptions which we make before we start our research. So how to
Right.
reduce that, zone of too much of assumption, I guess.
Absolutely. So that's where, that is why almost 25 minutes I spent on the hypothesis, because it's extremely important that you read about your topic thoroughly and understand what all the things that have already been done. And based on the observations, you come up with an attractive hypothesis that can be tested. Your hypothesis is based on strong observations that are already exist in the literature. And if you have methods to test your hypothesis, appropriate method, then the chances of success, meaning proving the hypothesis correct, is very, very high. But your hypothesis is shaky or based on assumptions that are not well supported, or your method that you use to test your hypothesis not appropriate, uh, your your success rate will go down again. So just to give an example, if you want to go to your friend who is living next door, meaning third house from your house, even if you have a car, you're going to go by walk. But that's the best way of reaching that place. Methods are something like that. Often we get, at times as a student, we have fascination for methods. For example, when I was a PhD student in early 90s, gene cloning and sequencing was considered to be something extraordinary. If I had sequenced the gene and then those days when you go to a conference, you will put the gel pictures there, the sequencing gel that will attract students to come and see. But that is nothing. It is the raw data that you are generating. It is not answering anything. So what I meant was you have to have a good questions and you have to choose appropriate methods to answer the question. And if the questions are for a very good hypothesis, your success rate is very, very high. Okay? That's what I would say. Therefore, you have to start, you have to read well, come up with very good hypothesis for a, hypothesis is nothing but a possible answer. So you have something that is not known. This is what you put as a question. As a possible answer, you develop an hypothesis. You say this is what is the possibility. And to say that it is indeed the case, you do experiment and you prove, yes, your hypothesis is correct. You prove that is right. And then it is no longer a hypothesis, it becomes a fact. But if you prove the hypothesis is not correct, if that is a long-held hypothesis, if you have proven that hypothesis is not correct, that itself is an answer, right? Think of it when if you have if you have not studied in your textbook that the world is in a globe, you wouldn't understand that. You would not believe that, right? You will say it's a flat surface. But then, if you see the textbook, the classic textbook in 20 years back, they'll talk about a ship coming. If you go to a seashore, if you see the ship, then you'll see the, there are ways, I don't want to waste time, but that is how, it has been told. Like, you prove something wrong if it is long-held hypothesis, that itself is, that is a failure, but you have proven that that hypothesis is wrong. That itself is amazing observation. So it all depends on the hypothesis. That's why I emphasize that you have to have a good hypothesis to begin with.
Yes, I do think so. Our next question is from Mrs. Chaitanya. She's asking how to choose the relevant journal. I guess this is what my understanding of the question is. At times, after writing a research paper, the next step is to find the right journal. At that stage, we might underestimate or we might overestimate. How to be reasonable about it?
Right. In fact, I would suggest the other way. When you have your results and methods ready, you should target journal. You should know which journal you are going to study first. Then you read about the instructions given for the journal and then write your research paper because there are journal-specific guidelines given. Now, to answer your question, like how would you choose which journal that you should consider for submitting your article? The best way is that, in your field, very similar kind of work with same amount of work or whatever it is, when was it published? Recently, was there anything that is published? And what are the journals that they are published? If you compare your work with that paper that is published, and if it is more or less very similar, then you would know that the chances of your work being submitted or considered, accepted in the journal is very, very high. So this is one way of looking at it. And second way is that aims and objectives, like each journal comes with certain scope, what do you call it? Read those scopes and see that your findings or your observations, your methods, the approaches that you have used fulfill the scope of the journal. If they do, then at least you look at some 5, 6, 7 papers there as to how much, like what is the data, like how much data is required for you to submit in that journal. How many figures, for example, what kind of methods, the impact of the work. If you read some 5, 6 and you find that is very similar, then you should consider. This is how you go about it. Then you have a higher chances of getting the paper accepted.
Great advice, sir. Our next question is from Avya. She's asking, what are the errors which are commonly seen or found in research papers written by students?
So I wouldn't call it as an error. I'm being a teacher, right? As a supervisor, many of those who are here in the panel, you know that your job is to mentor students, help them get better, right? So therefore, I wouldn't call it error, but then there are limitations, like you say that it is not mature enough to become good. One is that you need to know your limitation of your study, right? So you have to understand that there are questions and you are trying to answer, but your methods and approach or whatever you do, that would be something that will not answer everything that you try to answer. So everything has a limitation. You have to have limitations of your study first, and you have to know the strength of your study. And if you keep these two and then you write within this, you should not make statements that are superlative, meaning say that you use the yeast model and you're trying to say this is what happens in the human. That's something that you should not do. So you should know uh, within with the limitations how you can write. That is something that often that is missing. And second is that you have to have the order. Which one should be presented first? And which one should be presented next when you write result section? This is another thing that one has to be extremely careful. And these are the two things that most of the students lack, and with training they get.
Absolutely, sir. This would be our last question of the session. So Ameya is asking, have you ever faced writer's block? If you have, how did you overcome in your early days specifically? So
Oh,
they
yes.
want to be comfortable with that answer, sir. It would be inspiring for them.
I tell you, like when I joined for my PhD program in Banaras University, my teachers are all stalwarts, like they're all who's who in the field. And every week we used to have this what is called as lab seminars. Um, and my very first lab seminar was that 20 minutes, and I was only looking at the board. I did not look at anyone else. I'm just telling you, like that is how difficult it was. And same is the case. Like I could not face anyone. That's my confidence level then. And then when you write again something, you don't know where to start. It will be very interesting to know that this is the course that I have been offering to our own PhD students. We have a scientific communication course. This is a zero credit course. And when I was planning for this lectures, that's how we started, and I felt that I should give the NPTEL course as well. So many of this, then only I started looking at the literature whether somebody has done this kind of, how to write and things like that. Whatever I practice, like, that you start with methods and results. Why we say is that is the easiest thing to write. You know what you have done as a methodology, right? So it is there in your lab notebook. So easy for you to write. But if you start with introduction, it's extremely difficult. You don't know where to begin. So you'll lose your confidence. You start with what you can do easily. That is method. Easiest to do. Results is second one. Then by then you gain confidence, you go to introduction, discussion, and so on. So therefore, you'll be able to overcome that.
Yeah, well said, sir. We all have faced that time in our life. And with that, I would like to conclude the session. And I would, on behalf of the organizing team and Department of Microbiology, I would like to thank you. And especially, sir, wonderful presentation. And I think it's the right time to introduce such, topics in the curriculum also, because
Absolutely.
it's a skill and it requires the practice.
Absolutely.
And
I really recommend
yes.
like we have introduced this for all our masters and PhD students. This is a compulsory course here. There's no credit, but the curriculum. Yeah, that's something that I would recommend.
That would be great, and we would also be looking at it, to be introduced in our students' curriculum. And with that, a wonderful session, and it's a great experience to have you as a speaker. Thank you for being so kind in accepting our invitation, and it's a great pleasure to listen to you, sir. Thank you, and we are so
Thank you.
humbled and we are so
Thanks a lot.
grateful to you, sir.
I'm very happy to note that you consider this topic as one of the topics because normally people call me for such presentation. The first time I've been invited to give this kind of a talk. Thank you, and I'm sure.
And so we hope that you would be joining us in future also.
Yes.
I'll love to. Thank you very much.
So before you sign out, I just have one request to you and everybody here for virtual group photo.
Sure.
I would request people who are attending this to please switch on their camera so that photo can be taken, a screenshot can be taken. Janvi, are we ready?
Yes, ma'am. I'll take a screenshot at the count of 3. Would
Yeah, on
everyone
the
see?
count of 3. Yeah, let everybody come on the screen, please.
Okay, 1.
Janvi, you're not visible. Janvi, you yourself is not visible.
Ma'am, I'm not in a very presentable stage right now. I'm so sorry.
Doesn't matter. Come on.
Ma'am, I'm
Quickly.
seriously not in a presentable state. I'll just take a screen.
All right, okay. Okay.
3, 2, 1. Taken.
Thank you, Janvi. Thank you, sir. Thank you for joining
Thank
us,
you.
sir.
Thank you very much.
Thank you so much, sir.
Thank you, sir. Thank you so much. Such a wonderful talk.
He is signed out now.
Dr. Ipsa is here, Kriti, or not?
Ma'am, I can't see her right now. We'll just ask her.
Till now she is not here.
Participants, please stay with us. We're just trying to contact our next speaker. Maybe she's got some technical issue. So please hold on for a while.
While we are waiting, can I speak?
Sure, ma'am.
Yes, sure.
Yes, ma'am.
Most
Yes.
welcome.
Actually, I've always been very much concerned about students being able to write, and particularly technical articles, papers. And throughout my stay in Gargi, I have tried my best to promote that. And it is very, very important in today's scene that everybody is going into sort of that SMS language and shortcut language, and even in answer books, we find students at times using terms, I mean, words which are of sort of abbreviated and half the word and things like that. So I think in every class, for every teacher, it should be a kind of a responsibility that we at least spend one period in one session on talking about how to write well. And I have tried this throughout my tenure at Gargi, and I have found it very useful. There are lots of girls who have come back after years and told me that your sessions on how to write article, how an article is different from assignment, how an assignment is different from something which you want to publish in even a college magazine. That kind of differences are something which they need to discuss. We always expect so much from students, but do we really put in effort to guide them into writing like that? That is something I think should be given a thought. And I thought I'll just
Absolutely,
share
ma'am.
this with you while we are waiting for the next speaker. I must say that you have done a very good job of this whole colloquium, and ranging from I thought it's going to be highly microbiological and I would hardly understand. So with that feeling I had joined, but I just couldn't resist attending every session, because every session has a very different flavor to it, and I think that is something which goes a long way for deciding what to include in a 3 day workshop, because sustaining the interest of listeners for 3 days really means something. And I must appreciate all of you who have put in effort to do this.
Thank you,
Congratulations to
ma'am.
the whole team.
Thank
So
you.
appreciate.
Thank you.
So encouraging for us. Thank you, ma'am.
Thank you, ma'am. Thank you so much.
So Ipsa is joining in 2 minutes, she said. So she would be online
Okay.
anytime. Yeah.
Okay. Could we?
मैम एक चीज और पूछना था लाइक we also have this problem. We can also discuss here, but it's like most of the time I personally feel that students, like specifically when it comes to writing assignments and their practical records, most of them are copying from each other. They even don't want to try to write on their own. So how to change that attitude?
Can I answer that?
Yeah, मैम आपसे ही पूछ रही हूँ. You are the most experienced here.
No, yeah, actually, that is one thing we have to really. I noticed this for the first time, say about 10 years or so after I started teaching, that there was in all the files some sentence, and the say, like we tell them to write under the heading, so the under the heading of the principle there was a particular word which was technically wrong, and that was the last word in every file. And that is the time I realized that they're all copied, because we generally don't read the principle so thoroughly. We pay more attention to the readings and the analysis and all that. And since that time, I started telling students that this is a skill which you are learning. Don't call it making files. I just hate the word making files. You are not making paper and binding it into files. You are not making files. So I used to just all the time be after my students to resist this temptation, and many a times brought, I mean, called students who had copied from each other, and they would say, "Sir, ma'am, we did this experiment together. We were in the same group, so everything would be identical." But then I used to just
Is Ipsa is there?
understand. Is she here?
Hello.
Manpreet, Dr. Ipsa has joined.
Yes, I have.
Oh, hi, Ipsa.
Hi,
Waiting
Geeta
for you.
ma'am.
We'll continue our discussion some other time then. Welcome, ma'am. Welcome.
I don't know why you're calling me ma'am. I was your student.
So you're from Gagi?
Yes, and is that a surprise?
Yes.
Yes. Oh my God.
Oh my God.
Oh my God.
So
I
you
thought
are the
you called
alumni.
me. I thought you called me because I was I am an alumni, really.
Surprise.
Discovering it
It's a
now.
nice surprise. Nice surprise.
All right.
Welcome, Ipsa.
Well,
I
it's a
think
great to have you.
likewise.
You're from which
I was
school,
not like,
Ipsa?
Mr. Manpreet.
sorry, go on. Is this like a
Stream?
back room or are we live right now?
We are live.
We are live.
All right. I did zoology. Yeah.
Great. Dr. Indramani, I think we can
Yes.
start. Are you there?
Yes, I am here.
Yeah.
Can you
Yeah,
hear me?
yes, sir. Yes, sir. You are
So,
on.
Yes, sir.
so okay. So, in the continuation of today's session, good afternoon, everyone. And once again, I welcome to everyone in the second talk of the day. We have our today's next speaker, Dr. Ipsa Jain. Thank you, Dr. Jain, for your precious presence, and we welcome you.
Okay.
Now, I would like to invite Dr. Manpreet Kaur to introduce our speaker, Dr. Ipsa Jain, to the audience. Now, over to you, Dr. Manpreet.
Thank you, sir. We have with us today Dr. Ipsa Jain, a very talented science communicator and science illustrator. She is currently working at Vedantu as Instructional Designer Lead. Dr. Ipsa has her PhD from the Indian Institute of Science, where she worked on cancer cell migration and drug resistance. She has also worked as a postdoctoral fellow at Institute for Stem Cell Science and Regenerative Medicine, Bangalore, where she created sterilized representations of biology in the form of popular science books to generate public interest in science. Dr. Ipsa also runs her own freelancing brand called Ipsa Wonders, where she works with scientists, book publishers, and other kind of clients to create editorial illustrations for web pages, blogs, children's science books, schematics, and graphical abstracts for scientific papers. With great pleasure, I now invite Dr. Ipsa Jain to address the audience. Over to you, ma'am.
Thank you. It's a pleasure to be here. Um, I just want to first figure out how to share my slides. Just give me a second. Okay.
Sure, take your time.
Bottom you have this key
Yeah,
called
yeah,
share.
Just adjacent to, yeah.
yeah. Just
Can see your screen.
right.
It is visible.
All right. Excellent. Um, Yeah, so thanks for inviting me. It is a pleasure to always come back to college and again you with your own education and talk to the community there. So what I will try to do today is impress upon you, hopefully, that how visuals and drawing medium can be useful to scientific enterprise as a whole. And I'll just take it away, I suppose. And this session is meant to be interactive. So if you have pen, paper around you or pencil and paper around you, that'll be amazing because I might give you a tiny task where I asked you to scribble something and this is a judgment-free space. So no no praises for excellent drawing skills and no depreciation for not great drawing skills. So I'm someone who spent the majority of my life training to be a scientist, and I'm still one at core. But the tools that I use now have switched. I now use my pencil instead of a pipette. And as a proof of my credibility, I'm going to share a glimpse into my work. And as expected, like all of you here are a biologist, and because I'm also a biologist, the most of my work comes from, the inspiration for my work comes from biology. So this talk today will be part image display, part activity, part reading, and part scientific talk. All right. We as humans are curious. We want to try things out, to get distracted, and generally look like we are wasting time. Maybe we are wasting time right now. Sometimes we just want to see, to observe what is happening for the heck of it. To observe is to look at, listen to, touch, taste, or smell something, attending to the details of the resulting experience. We gain useful evidence simply by noticing what's going on around us. There is a very high concentration of nerves and finely tuned muscles in our palms and our fingers. As a result, we can perform very intricate tasks with our hands. The process of making knowledge or the process of making a material is a single hybrid affair in which the work of the head and the work of the hand form a complex whole. From 16th to 19th century, the illustrated scientific book remained the gold standard of scientific communication. Included detailed anatomical drawings by Vesuvius, anatomical tables and Euclidean diagrams by Copernicus, intricate microscopic drawings by Hooke, geometrical diagrams relating to fundamental laws of motion by Newton, uh, schematic mapping maps of the refractions of light rays, again by Newton, drawings of scientific instruments and tables systematically arranging the known universe of chemical elements and compounds in Biologia, geographical strata that reveals the Earth's history by Lyell, and floating microbes produced by fermentation by Pasteur. The 19th century also added a new visual modality to the scientific books and journal articles coming out from this endeavor: photographs. Some were taken with the aid of telescope or microscope. The The 20th century saw invention of image-producing research equipments like X-ray based analyzers, cloud chambers, particle accelerators, electron microscopes, satellite space telescopes, DNA analyzers, and so forth. In the late 20th century, the power of computer for the visual representation of large masses of data was harnessed. This has unleashed a wave of creative image making unprecedented in the history of scientific visualization. Image creation and analysis have always been at the heart of scientific discovery and dissemination. With built-in instrumentation, the role of hand, was it lost? Let's uncover. So what I'm going to do now is read out a passage, and my request to all of you is that try to visualize what I'm narrating. It'll be there on your screen as well, and just draw whatever you think is being described. Whenever we have a true cellular complex, an arrangement of cells in actual physical contact by means of a boundary film, we find this general principle in force. We must only bear in mind, for its perfect recognition, we must be able to view the object in a plane at right angles to the boundary walls. For example, in any ordinary section of a vegetable parenchyma, we recognize the appearance of a froth, precisely resembling that which we can construct by imprisoning a mass of soap bubbles in a narrow vessel with flat sides of glass. In both cases, we see the cell walls everywhere meeting by threes at an angle of 120 degrees, irrespective of the size of the individual cells, whose relative size, on the other hand, determines the curvature of the partition walls. On the surface of a honeycomb, we have precisely the same conjunction between cell and cell of three boundaries meeting at 120 degrees. In embryology, when we examine a segmenting egg of four or more segments, we find, in like manner, if not in all, that the same principle is still exemplified. The four segments do not meet in a common center, but each cell is in contact with the other two, with two others, and only three common walls meet at the normal angle of 120 degrees. So I just want to understand if this platform allows for people to show what they have drawn. Is that a possibility? No, it's being broadcasted. Oh, I can't hear you. You're not audible.
Maybe we can just show it like this,
Yes.
if it works.
Yes. Yeah, so I can't see attendees here, so I'm hoping that they can also show what they have done.
Yeah, they will have to. Yeah, so they will come on camera when they have to show.
Yes.
Somebody is ready already. Charu Agarwal is ready with a diagram.
All right. Yeah, so can we just like look at some 3 or 4 diagrams that some people might have drawn? If you can turn on your video camera and just share your drawing with us. Charu, I can't really see it all that clearly. All right. Okay. All right. Anyone else would like to share? No?
I guess it was too much of information for us to
Just
gather.
Okay.
Dr.
Manpreet, have you
Ipsa.
done something?
No, actually, I have a suggestion, ma'am. Could you just present that
Repeat.
question or present on the screen so that everyone can, it was really a big one. So,
All right.
yeah.
Can you see my screen now?
Not
Yes.
yet. It's
Not yet.
All right, just give me a second. Oh, yeah. All right.
Yeah,
So
thank you.
I'll reread it. Just give me a second. Okay, I'll just read the important part, I suppose. We recognize the appearance of a froth precisely resembling that which can be constructed by imprisoning a mass of soap bubbles in a narrow vessel with flat sides of glass. In both cases, we see cell walls meeting everywhere by threes at an angle of 120 degrees, irrespective of size of the individual cells, whose relative size, on the other hand, determines the curvature of partition walls. When we examine a segmenting egg of 4 or more segments, we find in like manner that the same principle is still exemplified. The 4 segments do not meet in a common center, but each cell is in contact with 2 others, and only 3 common boundary walls meet at a normal angle of 120 degrees. Okay, are people ready now? 2 more or 3 more? Okay. I can't see what you have drawn Manpreet. I can see what Kriti has drawn.
Geeta Mathur too has drawn.
Oh, I can't see. Is there a view setting that allows me to?
It's a
All right.
layout, yeah, layout on the upper side, right side.
Can you see mine?
Ma'am, she's trying to
Yes,
look at it.
yes,
You
yes.
know,
I can
Ipsa,
see
yeah.
yours, Geeta. Yeah. All right. Uh, okay. I'll just go back to sharing my screen and show you what it was. Right. Uh, So this is how a different set of people responded to the same passage in a different room on some other day. And this is what it really was. Some of you actually got it right. So each one of us are imagining objects and processes in a different way with all these clues and projecting them differently. This is based on our experiences with these tools and our prior knowledge of the subject matter. In his notebook, he begins with the phrase, I think, and ends not in a sentence but an illustration. A tree that represents the 3 key concepts descent, diversity, and extension. And this makes for the great tree of life. Visual displays are distinctively involved in scientific communication and in the very construction of scientific facts. Observations are fundamental base of all research methods. These beautiful images in Maria Sibylla Marian's Metamorphosis in Sectorium Surinamesium are the product of decades of meticulous observations of life cycles of insects. Most of her images were made from live or freshly preserved specimens. Marian shows moths laying eggs, caterpillars feeding on leaves. In order to create complex compositions containing a variety of organisms accurately depicted, she relied on individual studies made on small pieces of paper. When an animal or plant was copied onto the larger composition, she changed very little, preserving the posture, color, and other details of specimen or their relationship with other specimens in the drawing as sacrosanct as possible. Seeing, in some respect, is an art which must be learned. In his preface to Micrographia, Robert Hooke wrote that all that was required for studying nature was a sincere hand, a faithful eye to examine and to record the things themselves as they appeared. In the passage, Hooke implies that with his words and the pictures, he acted merely as an assistant to the microscope by recording whatever appeared through the lens of the instrument. This illustration of ant that he created at a scale which is many times its actual size highlights the insect's spiky limbs, brittle structure by silhouetting them against a stark white background, thereby effectively transforming a seemingly familiar ant into an exotic and mysterious alien creature. Hooke built this image from numerous observations made from multiple vantage points under varying lighting conditions and with lenses of differing powers. So, vision is not a mechanical recording of elements, but rather the apprehension of significant structural patterns. In this micrograph, darkly colored cells form close-knit clusters in an otherwise relatively uniformly stained tissue. To make these images, embryologist Saunders immersed living early stage chick embryos in a dye, the Nile blue sulfate, which is a vital dye. It colored cells without disrupting their normal processes. After washing, the pigment remained only in a particular group of cells. He at the time believed that these cells were precursors to the melanocytes. Things changed when he mapped these blue cells in a developing embryo. After staining and fixing tissue slices, he would trace the outer boundary of the tissue marked in these images by solid lines and the position of each blue cell onto a glass plate which is demarcated by dots. By lining up these models in sequence, he could see where the cluster of blue cells appear at each developmental stage. And he figured that these cells were not pigment cells, but in fact cells in the process of dying to part and articulate the digits. We need to show the real world, not lines and numbers. Oh, sorry. As we strive to create visual solutions for visual problems. The image here is painting of myoglobin, the first protein structure solved by X-ray crystallography. The painting was created by Irving Gies for Scientific American article titled The Three-Dimensional Structure of a Protein Molecule, written by John Kendrick. He created this painting by first photographing the physical model from different angles and then creating volume sketches and studies before painting this final version. A lot of refinements were made during the sketch step based on the feedback from the scientists concerned. And then he also made small adjustments to avoid structural overlapping. He might distort the protein a little bit, or he might choose a different viewing angle for a small part of this drawing or a different perspective. He wanted not to draw a protein exactly as it was, but to show how it worked. He called this process selective line and claimed that this was one of the special talents of a knowledgeable artist. A computer could draw a protein given the right set of coordinates, but who would tell it what to draw? In 1968, Waddington tells us science is something more than a collection of conceptual and practical results. It is also an activity, and its practice involves, as a very important part, the exercise of the faculties of insightful perception of the natural phenomena and of the imaginative creation of new concepts. The image on the left is a top view of the epigenetic landscape where the path followed by a ball represents developmental trajectory. The valley represents different alternative paths to depict different developmental potentials. For example, differentiation leading to various cell types. On the right is the underside of such a surface, where he illustrated the idea that genes can change the landscape during the evolution. So the pegs at the bottom represent the genes acting on the landscape by tugging on the intertwined web of ropes and thereby changing the topography of the valleys and ridges above. This image created by Waddington does this very well. It stimulates visual thought. To paraphrase Waddington himself, his artful representation helps to loosen the joints of the scientist's imagination, creating a space for associative play required to introduce new concepts in theory formation. The ball in Waddington landscape remains same all over the time as it rolls down the valley. Now, this is a problem. So, artist Gemma Anderson and the collaborating scientist imagined overcoming this limitation by replacing the ball moving through this landscape by,
It's a
yeah,
sorry to interrupt. That pop-up message which is coming on your screen. Can you please hide it? It's distracting.
sorry.
The hair which you have here, that stop sharing and hide
Oh, I see.
message which is coming there.
I don't know. Okay, let me see. Okay.
Yeah, yeah. Thank you.
Okay. And anyone, if has any questions, please ask. Otherwise, this will just seem like a monologue. Um, uh, Sorry about that. So, Isomorphogenesis is a drawing based series in which artist Gemma explores the potential of representing morphologies as a dynamic and formative process. The one you see on the right is her earlier work where she imagined change from an embryo to an adult as a series of overlapping drawings. And this is what she tries to superimpose on the Waddington's landscape as well. The resulting experimental image reduces the level of abstraction of Waddington's representation by explicitly showing the trajectory of morphological change through the valleys of landscape. Creativity, which is the formation of new associations and concepts, requires playful abandoning of preconceived notions. To be creative, we must let our minds wander. She says hand drawing must not be allowed to disappear. And she says this not because of its use in scientific illustration, but because of its potential to form insight and understanding, and to share this understanding, and to enhance creativity amongst artists and scientists alike. So now we are going to try and be a little playful. I have some words in the upcoming slides, and my request to you is that I will have a timer on my phone for 1 minute, and you have to draw the first thing that comes to your mind when you hear that word or you see that word. All right. I'm just figuring out my stopwatch. All right. The first word is warm. All right, time up. Now we'll do another word, and we'll all share our drawings at the end of drawing all both the words or phrases. Oops. Glowing warm. All right, time's up. I'm going to stop sharing again so that we can see what you have drawn. All right, whoever wants to turn on their cameras and share the doodles they came up with.
This is, I drew a cup of water for worm. And for glowing worm, I know jellyfish is not a worm, but I took the glowing wala part. So that's why I drew jellyfish.
Okay, anyone else would like to share?
This is from my side. This is a glass of water and sun is really shining and we are not happy with that.
Okay.
Wow.
I
All
drew
right.
cap and muffler in worm and fire and glowing worm.
Okay, I can't see what Geeta ma'am is trying to show. Like I see what she's holding up, but is there a way for it to come to the main screen? Like
I have drawn one cup with the marks for steam coming out, and a bowl of soup with a spoon, a sweater, and a cap. And for glowing worm, I have drawn a segmented worm and shown glow all around it with small lines.
Cool. Okay. Yes, I can see those two drawings from Shashi and Prithvi. All right.
For worm, I've just drawn that smoke kind of thing which appears when you have a cup of tea. And this is warm and just glowing. I mean, there's no way to show it, but just with the color dots we are trying to show.
All
I've
right.
added few lines as if we are drawing the bulb for the glow.
Glowing bulb, diya for glowing worm.
Okay.
Where's the worm? That's been very
When you will come around this diya na.
He will burn, he will not glow.
Okay, and
That's
why
more like
are
a
the
poetry, no?
students not participating? Why are they so shy? Come on, take the lead of your teachers and show us what you've drawn.
Ankita, Anshul, you guys are so creative. What has happened? Priya? Bhavya, Arushi, come on.
I'm also going to think of this as a break before I resume to the next part of my talk, which can be slightly technical. So if you have any questions about images and sign, like this is also a place to ask. I mean, is this meant to be interactive? I'm sorry, I didn't ask you.
Well, we have no such norms for that. It's
Okay.
open.
Okay. All right. People aren't really interested in sharing, I suppose, or asking. I'll move on then. All right. Renault collaborators once said drawing schematics. Drawings can provide space-based visualization, which could never be photographed and definitely never be schematized. So together, we wanted to make drawings of cargo being carried across the cell. Is it safe for me to assume that people here know a little bit about molecular biology? If any of the teachers can respond.
Yes.
Absolutely, Ipsa.
Yeah, all right. Thank you. So, what we just wanted to draw was like how a cargo moves from the center of the cell to the periphery of the cell and how it moves from periphery of the cell to to the core of the cell. And to that effect, we starting studying dynein, which is the motor involved there. You don't need to necessarily know a lot about dynein, except that it works on this organelle called microtubule for the context of this conversation. Uh, so the first thing that I did was I looked at the structure of this molecule and I started making sketches of this molecule based on what I could find, and then try to understand how this molecule actually looks like. And so I first made a lot of rough sketches, and then I tried to make a lot of fair sketches from different point of views, And as I started understanding about the form, the next thing I wanted to do with this was like, so we know from literature that this motor really works as dimers. So like the two of them work together. And so my intention then now became as to, how do they, sort of tag together and move. So I try to understand their relationship with each other. And through that effect, I read a lot of literature that was present around by, based on in vitro studies, in cellular studies, and whatever else I could find. And it almost became like a puzzle solving exercise where could get different bits of information from different parts of the literature, and then you know doing this activity made me realize that what are the things that I do not know. Like I did not know how to draw this line, or I did not know how to show this molecule, which meant that that's the information I had to look up and then do more research. So this was quite an iterative process for me, where I drew multiple models, and again from different points of view, from like from the organismic point of view. Let's say if you wanted to do dorsal and ventral, so similarly how the molecule will look if it's traveling from this side to this side, and how it would look if it's walking from the other side. And this basically led me to a lot of questions. I figured out not only, and this was the final drawing that I could come up with. And this drawing is comprehensive in the way that, like, at the moment that it was created, it encompasses all the available literature to the best possible extent. And it shows not only what we know, it also indicates what we know. So what we don't know. So the things that we don't know very well are, I've drawn them with dotted and loose lines, and they're not as firm in the stroke quality as other parts of this drawing are. So after I figured out how they, tag with each other, I wanted to understand how they work with each other. And again, I did the same exercise where I looked at different pieces of data. Don't feel intimidated by this data on the other side. But like if you look at what my sketch is on the other side, I basically tried different permutations of and combinations. Like, oh, so this paper says that it works. In inchworm condition or head over head. Let me just tell you what is inchworm and what is head on head. So inchworm is like if I move this much and the other guy also lags behind, or I move 4 steps, the other guy maybe does 5 steps, maybe does 3 steps, 2 steps. There is no real relationship. Like it's just that one moves first, the second moves later. One moves first, and the second moves later. And head on head is more like how we walk left, right, left, right. So there is a sense of how. So there were different kinds of literature which said that either of these type of works is possible, and I just simply explored them on piece of paper to figure out how that would look like. And by that time, I had one more piece of data available. So literally at the moment that I was trying to draw this and research about this, another electron microscopy based cryo-electron microscopy based data came into light, which kind of showed what's the starting point for this such a dimer. So once I had the starting point fixed, I could then create multiple walk combinations. And what I'm going to do now is quickly show you these different walk possibilities. So 1, 2, 3, 4. 1, 2, 3, 4. And if you notice, like the number of steps I have are equal, but in different permutations of how the walk has happened, the amount of distance that these two molecules together have been able to cover is different. Like, I hope you can appreciate that. Like 1, 2, 3. So this black line, which is towards the left, sort of indicates the distance covered, whereas the line on the right is the starting point, which happens to be the same for all the instances. So this essentially kind of became like an exercise in simulation where drawing could be used to generate a hypothesis. And uh, so at the end of this whole exercise, I came up with a lot of questions, and these questions then still remain unanswered. We asked questions about how does the cargo move when the motor moves? Do both the motors have to move, molecules have to move before the cargo moves, or can the movement of one motor itself push the cargo forward? This is something that we have zero clarity on. And all of these questions were possible because of this drawing exercise. So, what is really cool about drawing science is that it's free from the constraints of experimental feasibility. So, in an experiment, you will ask while you're trying to develop an experiment, you will sort of strategize based on what's the available tool and can you, but this one relies on your imagination, so there is a possibility to create and think beyond. So now we're going to do another activity, and I'm hoping that the students will participate this time. Right, so what we're going to do is I'm going to show you an image, and we'll call that observation at time 0. And the task at hand is that you have to think what is going to happen to it next. And that time point t, you are at liberty to define what that time point is. It could be 1 second later, it could be 1 minute later, it could be 1 day later, it could be 1 century later, or it could be 1 eon later. That's entirely up to you. So I'm going to show the image for around 1 minute, and then give you space to think for another minute, and then space to think and draw for another minute, and then we'll just do the exercise of sharing with each other again. Right. So here's the image. Just ignore the email ID that's there at the bottom. Yeah.
Second slide about the whole model itself, where you will realize the resilience of the model in the diversity. Now has an established chain or channel for to reach out to genes. Even someone else can leverage it at the moment. And it has become a model for scale and broadcast. So this was something that he mentioned in his talk. He says that scale broadcast for when it comes to such initiatives. The third thing, I mentioned this again just for security, because they could secure funds, it also meant they could collaborate with a lot more people. This is again one of the interviews with Isha. And this is what Raghu specifically mentioned, that because they could establish this model, actually they know this model. They had now have a
All right, so 1 minute for observation is gone, so hopefully you can start drawing now. And I'm going to give you a minute to take a snapshot if you want to take a snapshot. I'm going to stop sharing my screen in just a moment. All right. And another 1 minute timer. Oh, you've already drawn. All right. Okay. Yes, I can see you're drawing. All right. Anyone else would like to share? I'm really hoping that some students share this time around. I can see your drawing, Manpreet. Should I just like randomly take people's name? But I don't like to pick out and pick on people.
Actually, some of the students have network issues, so even if they want, they are not able to show it because this platform is really not very,
I see.
user friendly. That is another issue.
Right,
There
right.
is another Gargi Samanta. She's also showing something.
Yes, I can see.
Now, some of them are writing in the chat box also that the problem is now showing.
I see, I see. I see, I see. You'll have to excuse me for a minute. My patch keys, I need to put my charger. Sorry, just hold on. That gives you time to draw, everyone. Huh? No?
Priti, meanwhile, you can make an announcement for feedback form.
Okay, it's a request to everyone. Now, please try to fill in the feedback form. That would be really great if you can. We've dropped the link for it in the chat box.
Right. Are there any people who would like to share their drawings? Okay, continue. Um, um, the points that I wanted to bring out really from this exercise was that different people usually tend to come up with different ideas as to what will happen to this. And if you think about these, all of those ideas become testable hypotheses for what can happen to this entity here. So some smart person said hypothesis that may be worth pursuing may be realized verbally or visually. The latter may be described rather than depicted. Uh, so moving on to the next sort of segment. Um, I asked students, both students from science, maybe some of them, including your friends from Deen Dayal Upadhyay College, science students and law students to, draw cell, whatever they remembered of a cell, and these are some of the representative images. And like if you notice it carefully, the images on the left and the right have some relationship. They all speak of a boundary, a central nuclei, and some specs within those things, except those specs are defined in among science students and not so defined amongst the lawyers who ended up drawing this. Um, so the and what is a common theme, of course, here is that most of these drawings show cross-sectional view of the cell, whatever be the level of details. And a lot of science teachers, particularly from mention how students think of cell as a flat entity, like it's a sunny side up, and if the cell was to slide, then things will just fall out of it. The idea that cell is actually a 3D entity which has a volume does not come across through images like this. So I wanted to, sort of take that up as a visualization challenge and think of what I could do with it. So I started making a series of scenes about cells, and I came up with this visual metaphor. So I thought, like, let's slice up a familiar object, which is orange in this case, and then slice up another cell, which was tetrahymena in this case. And as we look through a slice of orange, we also look through a slice of the tetrahymena cell. And what happened when I created this visual metaphor and then I printed it and presented it at different places, including conferences and comics festival, is so you can imagine that the audience in conferences was science students and or scientists in training, whereas people at comics festival were people who may or may not necessarily have a love affair with science. So. Irrespective of those backgrounds, people were able to identify relationships. Like people who did not know that this organelle is called mitochondria were able to identify that how mitochondria is mainly present towards the periphery of the cell. And it seemed like whatever those structures are, cilia and the mitochondria seem to have some relationship. So there was an intuitive relationship building because this visual metaphor used actual scientific data. So the drawings that you see here for the cross section were in fact heavily dependent on electron microscopy data for tetrahymena available. And there were people who could appreciate the cells, so like how distribution of organelles across different cross sections were different, and how the cross section diameter widened and then tapered again. So all these clues, without knowing the jargon, was something that was the information that we could gather by looking at all of these images together. So I'll conclude, I mean, not, I just conclude this bit by saying we see, but we do not see. We use our eyes, but our gaze is glancing, frivolously considering its object. We see the signs, but not their meanings. We are not blinded, but we have blinders. So with their scientific argument, these two gentlemen turned the X-ray diffraction photograph of DNA taken by Rosalind Franklin into a possible stereochemical realization. The model further applies to any DNA molecule and not the one that was just used for the study. Each of its realization stands for the rest. The molecule structures suggest a scheme of replication, a shift from description to explanation. Conceptually, it was easy to visualize how a single chain would be the template for synthesis of a chain with complementary sequences. While no one would claim that images are integral to all scientific thinking, it seems indisputable that scientific thinking, sometimes of the highest order, occurs in the absence of words. And when probing into the unknown, drawing is an ideal tool because drawing is thinking, pointing. It makes things present and puts them at a distance. It's a mode of inquiry. Drawing can represent the seen and present the unseen, the known and the unknown. With that, I'd like to thank you for your attention. These are some of the references that I've used for images and readings throughout the talk if you like to read up further. And thank you for your patient attendance. And I'd like to thank my collaborators in my lab where I did the dining work. And a lot of other work has happened in with a lot of collaborative spirit. If you would like to look me up, I'm easily available and accessible on social media. You can check out my website as well to learn more about illustration. And I'll happily take questions. I'm hoping that people will ask questions. Yeah.
Thank you so much, Dr. Jain, for such a great and wonderful talk. It's truly enlightened us. Now I would like to invite Tanvir, our student, to moderate question and answer session. Over to you, Tanvir. Tanvir, can you hear me?
Am I audible?
Yeah, you are audible.
Good evening, ma'am.
Please go ahead.
Thank you for so wonderfully enlightening us on the topic. So we have few questions. The first one is, ma'am, so like when students take up science as a degree course, the people often want to pursue research or teaching profession. So can you please guide like the pathway about how can students pursue scientific illustration and communication as a higher educational degree course or a career?
Right. So, I mean, this is an easier question to answer at your age. I made a pivot after I finished my PhD, which was sort of, I think, the harder thing to do. So, there are multiple ways that you can think about this. So, there are some professional courses on science illustration available across the, the globe. Unfortunately, none in India. So there are a few good courses in Canada, UK, some in Europe, US, of course, and Australia, which offer excellent courses on science illustrations. The other thing you can do is, if that kind of education is not something that you can afford or spend your time on, is you can also try to go to fine arts or illustration courses at places like NID or Srishti or IDAC, Mumbai, or any of those places which offer excellent programs in illustration. And with your scientific training and your skills developed at the illustration course, you will be able to combine the two and then work towards being a science illustrator. India right now doesn't have a formal program as to how scientific illustration, who can do scientific illustration. In US and Europe, there are like specific guilds where you have to register to be working as a scientific illustrator or as a medical illustrator. But in India, it's right now not catered, which is also excellent in the sense that it allows anyone the opportunity to do that. And I would say that if you are somebody who does not even want to take a formal education, like I didn't, the one way to go about it would be to practice drawing on your own. I mean, I suppose all of us, I mean, I don't know, I'm hoping that that's true for microbio also, where you do a lot of observation based drawings. I mean, you look at the slides and then you have to kind of draw what you so we tend to rely on reference images for that sort of drawing. And if you just develop this eye and coordination rather, where when you look at something through the microscope and then you are able to recapitulate that in your drawing, that would really add to your existing skill set. And you can then independently learn drawing, as a side course or something that you do from Skillshare, Coursera, or any of these online learning platforms. And the next thing, once you have any of those parts to have picked up illustration skills, would be to build an online portfolio. And once you have this online portfolio, you can reach out to your clients. And these clients could be anywhere from scientists to publishing houses, or people who make books, and or people who make journals like the Nature Publishing Group, or any of those kinds of outlets. Yeah.
Thank you, ma'am. Next question is there any software to draw the picture?
I mean, so of course there are softwares which allow you to develop these figures, but I think the first thing that you need to learn is what do you want to draw or what you should draw. I mean, software wise, and I think on freely accessible ones, there is GIMP and Inkscape. And if you're looking at paid softwares, there is Adobe Illustrator or Adobe Photoshop, of course, and then if you use iPad or something like that, then there's also Procreate. Uh, but I have seen so far that knowing these softwares does not necessarily translate to knowing how to do scientific illustrations. So first, you know what is it that you want to tell using that skill set. So thinking takes a longer time to develop than skill set. Skill set is something that you can develop on the go. Yeah.
Thank you, ma'am. The next question is, are the terms science communication and scientific communication same?
I'm not sure if I'm necessarily the right person to answer this question, and also I'm not sure if there is a agreement amongst the scientific community about these terms. But how I is scientific communication is very much limited to when you are communicating with the scientific peers itself. Like if it's a scientist talking to scientists, which can be either via posters that they present during conferences or talks they give during conferences or reviews they write in journals or the actual scientific articles they write in journals. So I think of that as scientific communication, and science communication, I think, is an open term which talks about taking accessibility of science to larger audiences. But I think this is a more personal interpretation and not necessarily a formal interpretation. Yeah.
Thank you, ma'am. Can you please elaborate on one of the many aspects of scientific illustration, that is medical illustration?
Again, I'm not sure if I'm really the right person to answer this question, but I do have a friend who is a formally trained medical illustrator. Her name is Tejaswini Padma. So if you want to reach out to her, I'm sure she'll be happy to help you out. But specifically to be a medical illustrator, you need to do a formal course. Like the kind of illustrations I do, I don't necessarily need to have any formal course to be able to do the kind of work I do. But medical illustration is one field where training or formal training is a must, because you do end up working with doctors and cadavers and all that sort of stuff. So it's essential for the training to know what the theology is in the most deep way possible. Yeah.
Okay, ma'am. The next one is, how promising you see scientific illustrations in context to job prospects in India currently?
Oh, well, that's the harder question to answer. But all I can say is that the community is growing, and the thing is with such jobs, which such unique jobs, is that unless there is not a critical mass of people who are doing these kinds of jobs, people who make decisions, people who make policies will not take you seriously. Like scientific writing at a point of time was a rare job, but now it's not. A lot of institutes hire scientific writers on a regular basis, but the same cannot be said of scientific illustrators. So I like to think that we're waiting for that critical mass for to happen to make that noise and say like we need proper placements. Right now, I would say that one has to end up diversifying their sources of income and to be able to build a sustainable career. Um, and there are no jobs specifically. It works like gig economy, and I think your generation really has to be okay with the idea of a gig economy, because going on the idea of jobs itself is going to be scant, and we'll mostly be working in the gig model. Yeah.
So, ma'am, we have one last question. It is, what is the role of scientific illustration in the field of forensic science and fossil studies?
Yeah, so those are the kind of illustrators which end up coming from the medical illustration field, especially the ones in forensic, the ones who end up joining crime divisions and uh, forensic branches. They are usually medical illustrators, and there is a lot of like possibility of those kind of works across the globe. But again, unfortunately, not in India. So one really has to step outside the country to be able to do those things. And again, the even across the globe, the jobs are not a lot. And while there are a lot of illustrators, so you have to be really good and in that pool of competition to be able to make it.
Thank you, ma'am. Ma'am, there's another question from a teacher, Geeta ma'am. She's asking, what was your interpretation of you made us do in the beginning?
So the one that was about the growth and form, that was basically to say that there is a certain distance between words and what those words make us imagine. So the reality and imagination might have a difference when we are describing something using text. And the idea of that activity is that you realize how and why images are important in science. And the second activity where we did the worm and the glowing worm was to, really comment on how when we think of something like worm, we end up iconic symbols like the sun or a hot cup of tea or uh, a thermometer or something like that. Like we rely on these symbols for showing an abstract idea. And through our education and through our culture, we have somehow developed a uniform vocabulary, uniform visual vocabulary for those abstract ideas. And the context of creativity, it might be useful to think beyond those symbols. And with glowing worm, the idea was that worm being a tangible example, it might be easier for us to think of a glowing worm as something that it's just that worm that came to your mind or my mind would be different, but at least it'll be a worm that we know and can recognize. Yeah.
Thank you so much, ma'am, for so patiently answering all the questions. That's all the questions we have now. Thank you, ma'am.
All right, I'll take your leave. Bye-bye. It's a pleasure.
1 second, Ipsa.
All
1
right.
minute, please.
Yeah, sure.
Thank you,
Your
Ganesh.
video is not visible.
My video?
Yes.
I can see my video on this chat window.
Let me confirm with some. Shashi ma'am, are you able to see Ipsa's video? No, we are not able to see you, Ipsa.
Okay, just let me try that again. Can you see me now?
Not yet.
Okay, this is kind of weird because I can really see myself. It's a picture-in-picture for whatever reason.
Maybe there's a bandwidth problem. It could be one of the reasons. So I'm not sure. But
Otherwise, on the top, you have this layout. No? Just try to change the layout.
I don't really see a layout icon actually.
No. If you see at the corner of this is
Right
written
side.
due to low bandwidth or local computer conditions, video is not currently available.
I see.
Oh, okay.
So it's written there in the corner. If you see this Ipsa's corner, Ipsa corner.
All right. So
So
there's
there
a
is a
network
symbol
issue.
over here, there. So it tells that.
So nothing can be done much about it, I guess. Okay, Ipsa, actually we wanted to have a group photo, virtual group picture.
I
But
see.
I guess it would be with your name only now. It can be done. I don't know.
No problem. That's fine.
Yeah,
All of that, yeah.
yeah. So, Janvi, are you there online?
Yes, ma'am.
Yeah, please.
I can send you my screenshot because I can see myself.
Please do that, Ipsa. That would be wonderful. Nice to see you, Janvi.
Could everyone please switch on their cameras?
Gauri, Bhavya, Devanshi, Deepa, quickly.
And you were showing some pictures, so at least photo to खींचा लो.
सही है.
Janvi, when you're ready, just tell us, right? On the count of 3, you can take the picture.
3, 2, 1. Done.
Thank you, Janvi.
Thank you.
And Ipsa, thank you so much for joining us. And your slides were beautiful, and it was treat to watch. Great experience. Wonderful presentation.
Thank you so much.
And of course, science without diagrams cannot be imagined. And
I'm hoping that the
looking
people
at
who
your yes.
sorry. I'm hoping that the students who watched this exercise will now look at their file making exercises a little more dearly than they
Yes,
did
and
before.
it can now be appreciated that whatever diagrams they look into in their textbooks, it requires a lot of imagination and that connection between what is to be communicated and how you actually in 3D you draw that. Otherwise, it's actually very difficult to convey what actually we are talking about. So it takes a lot of, I guess, a lot of patience and a lot of, I don't know what to say. You need to have that vision about it. I can never imagine myself drawing such complicated. I mean, hats off to you, Ipsa. It's great work. Unusual, alternative, and absolutely wonderful. So Ipsa Wonders is correctly named. Yeah.
Ipsa Wonders. Yeah. So you have to be very, very imaginative and creative.
And specific
I
about
think so.
all the minute details while you're drawing it.
Yeah.
Absolutely.
Of course, you have to be a little scientific also. Yeah, true.
Yeah, well,
You have to gather a lot of data for that.
So Ipsa, and we hope that you would join us again in the future. And it was wonderful to have you, and a cute little potted plant would reach you as a token of
I think it
gratitude.
already did.
Oh my God, it's delivered. It's a little embarrassing.
All right. Thank you
So
so
please
much for the
accept
gift.
that with lots of love to you from
Thank you.
Department of Microbiology. Thank you. It's been wonderful to have you. Thank you.
Ipsa, I also want to add that please share a picture of yours with carrying that particular plant with you. Please share
Oh,
the
it's
picture with
my
us.
in-laws place. When I get to it, I will surely do. Yeah.
Yeah, thank you.
Thank you, Ipsa. Thank you.
Bye. Have a
Bye.
good day.
Thank
Thank
you.
you,
Day.
Ipsa.
Thank you. Welcome. Bye.
Thank you, Tanveer. Thank you, Dr. Jain, for sharing valuable knowledge with us. Thank you so much. Now, I would like to invite Dr. Chaturvedi, coordinator of colloquium, to formally give the vote of thanks. Over to you,
Thank you,
Dr.
sir. Thank you, sir. So it's now time to formally conclude our 3-day colloquium in Kindle. I, Dr. Kriti, on behalf of Department of Microbiology and entire supporting team in the capacity of coordinator, is privileged to propose a vote of thanks. First and foremost, I would like to extend my sincere gratitude to the distinguished speakers at Enkindle, Dr. Christine Moisel Enger, Dr. Tony Gutierrez, Mrs. Vani Murthy, Mr. Akshay Jain, Professor Ganesh, and Dr. Ipsa Jain. Their presence at our colloquium has been encouraging and motivating for all of us. We are overwhelmed by their presence, to have such eminent speakers at our event. I would like to express my profound gratitude to our principal, Dr. Promila Kumar, for being so appreciative and giving us this great platform to organize our event. I would like to thank our teacher in charge, Dr. Shashi Chavla, for giving me this opportunity, for her constant support and encouragement. And her positive, uplifting attitude is worth appreciating and being the driving force for us. I also extend my hearty thanks to the conveners, Dr. Manpreet, Dr. Indramani, for all the support and help throughout the event. I'm grateful to our very motivated and dedicated students organizing committee for their enormous support in executing the event so efficiently and successfully, especially Mansi, Anshul, Janvi, Vanshika, Shubham Priya, Ishika, Tanveer, Pranshi, Ignita, Priyanka, Khushi, and Garima. And please pardon me if I missed somebody. And I'm appreciative of all our students for their active participation. Most importantly, I extend my gratitude to the wonderful audience affiliated to different universities and academic institutes, and also faculty members from our own college and other colleges of DU. And I would like to thank Mr. Anuj Bhardwaj for all his support. He's been a very dedicated technical support we have got. So thank you, Anuj, for all the support in executing and broadcasting our event so smoothly. So once again, I extend my gratitude to our esteemed speakers, Principal Dr. Pramila Kumar, Dr. Shashi Chawla, Dr. Manpreet, Dr. Indramani, our students, Mr. Anuj Bhardwaj, faculty members, and audience for making Enkindle, a 3-day colloquium, a great success. Thank you for being with us. We shall be signing off now. Stay safe and stay healthy. Shashi ma'am, would you like to add something?
Thank you to everyone. I will just say
Thank
that.
you, everyone.
Thank
Thank
you.
you.
Thank you for those 3 days.
Thank you, everybody.
I think we can sign off.
Yes.
Yes. Archana ma'am is left. And thank you, Geeta ma'am and Archana ma'am, specifically.
Yeah, so I think she's there.
It was a pleasure being here in this very well organized activity and very useful too. Thank you all
Thank
again.
you. Thank you,
Thank you,
ma'am.
Thank you,
ma'am.
ma'am.
Thank you.
Thank you so much.
Okay, bye, everybody. Anuj, thank you so much.
Bye bye.